Good morning and welcome. I'd like to welcome you to this webinar on dermatophytes hosted by R-Biopharm Australia and presented by Josh Griffin with special guest Darren Jardine from St Vincent's Hospital, Melbourne, and subject expert matter Misha Hashmi from R-Biopharm. We honour the Gadigal people of the Eora Nation, the traditional custodians of this region in which R-Biopharm Australia is based. For more than 65,000 years, Aboriginal and Torres Strait Islander people have created knowledge as Australia's first scientists, technologists, engineers and mathematicians. This long-standing STEM knowledge is reflected in the active care and management of country. I'd like to take this opportunity to introduce R-Biopharm Australia. We are manufacturers of in vitro diagnostics, reagents and robotic systems for molecular-based syndromic testing. Specialising in highly multiplex PCR, our method enables more informed diagnostics decisions in human health, animal diagnostics and applied testing. With one of the most comprehensive testing panel suites in the industry, our products address testing needs within the areas of respiratory, enteric, STI, antimicrobial resistance, dermatophytes, among many more. Today, the R-Biopharm Group supply to laboratories and hospitals around the world from our headquarters in Germany and offices in New Zealand, United States and United Kingdom, with a mission to improve patient outcomes through diagnostics innovation. Just to let you know, the session is being recorded. Following the presentation, there will be a Q&A. Please feel free to leave any questions in the Q&A chat. I'd like to take this opportunity to thank and introduce Joshua Griffin for joining us as the presenter today, sharing his knowledge and expertise. Joshua is a Grade 2 medical scientist at St Vincent's Hospital in Melbourne, specialising in mycology with a keen interest in molecular testing. Josh has successfully implemented dermatophyte PCR in the laboratory, allowing for faster turnaround times, improved laboratory workflow and better patient outcomes. Joining us as a special guest is Darren Jardine from St Vincent's Hospital Pathology. Darren is the principal scientist of the microbiology department. His interests lie in development and implementation of molecular diagnostic tests for detecting human pathogens. We also have joining us subject expert matter Misha Hashmi, a product manager and molecular biologist with experience spanning clinical trials, research and development and molecular diagnostics. Misha brings a holistic perspective to human diagnostics, combining scientific expertise with end-to-end product development and commercial strategy. I'd like to hand over now to Misha to start the presentation.
Thanks, Olivia. I'm just going to quickly give everybody an overview of the current dermatophytes portfolio offered by R-Biopharm Australia. We have the Retoplex dermatophytes and other fungi 12-well panel, which is IVD marked in multiple regions, Australia, New Zealand, Europe, and the UK. And as is in the name, it targets both dermatophytes and other fungi. Its targets include epidermophyte influcosum, Microsporum canis, genus level primers for Microsporum, Nenetsia gypsia, genus level primers for Scopiopsis, genus level primers for Trichophyton, all the species, in addition to the species or complex level primers for Trichophyton rubrum and Trichophyton mentagrophytes. In the other fungi section, this panel offers genus level primers for Aspergillus, Candida albicans, Candida parapsilosis, and Leucosiomyces glabratus, and also Myrzema guermondii. Launched in 2020, this panel was received well. There was a lot of positive reactions from a lot of laboratories, and many took up this solution that we offered alongside fungal microscopy. However, we did see a lot of customer feedback in the field that Aspergillus genus level primers, Candida parapsilosis, Candida glabrata, and also Myrzema guermondii, these targets lack clinical utility due to their commensal nature. As we developed this panel, we ensured that there was full automation solutions and also compatibility with our RetaPrep tissue digestion buffer kit for downstream nucleic acid extraction. And this is where I'll hand over to Josh, who's now going to bring us into the current, the launch of our dermatophytes A panel. Over to you, Josh.
All right. Thanks, Misha. Good morning, everyone. Just give me one sec to get this going. Oops. So, as Misha said, my name's Josh and I work as a grade 2 medical scientist over at St. Vincent's Hospital in cold old Melbourne. Today I'm here to talk to you in regards to our validation that we did on behalf of R-Biopharm in validating their new dermatophytes A12 or assay, which promises to distinguish Trichophyton indotineae from Trichophyton mentagrophytes. All right, so I'll start off by giving a brief background on some of the common dermatophytes that you might encounter, with a particular dive into the Trichophyton genus, including the species Trichophyton mentagrophytes and Trichophyton indotineae. I'll then give a brief overview on R-Biopharm's assays as a whole, how they work, and the current dermatophytes and other fungi assay that is on the market, and then into our work validating the new dermatophytes A12 or assay. So, what is a dermatophyte? They're a genus of pathogenic filamentous fungi that commonly cause skin, hair, and nail infections in humans and animals. They digest and obtain their nutrients from keratin, a protein found in hair, skin, and nail. Generally, 20-25% of the human population each year are infected with a dermatophyte. And these infections are commonly referred to as ringworm or tinea and are a result of the host cell reaction to the fungus cell wall during its keratin digestive process. So what that looks like on a human is you get that tinea ringworm kind of looking scaly rash on skin. You can get onychomycosis on toenail and fingernails, and then scalp and hair invasion infections. So what are some of the common dermatophytes you might encounter? You've got your Trichophyton species in the top left-hand corner there, Epidermophyton flocosum, Microsporum species, and Nunasia species. Like Mr. said, these 4 species are currently available as gene targets on the existing dermatophytes and other fungi 12O assay. And they make up about roughly 99% of all the dermatophyte infections that we see within our laboratory. We're now going to talk more in depth about the species, the Trichophyton species genus. Then some common descriptions within the genus, including their macroscopic and microscopic characteristics. So the genus contains about 16 different species, which has been whittled down from 22 since the onset of DNA sequencing, showing that some of those species were actually deserve their own speciation. This Trichophyton species can either be zoophilic or anthropophilic in nature, with zoophilic Trichophyton species liking animal hosts, and then the anthropophilic liking human hosts. They are characterized morphologically by the development of microconidia and macroconidia, which are the small asexual spores of the fungus. The most common species of Trichophyton you will encounter are rubrum, mentagrophytes, interdigitale, pseudonancy, tonsurans, and violaceum, each causing various forms of infection within humans. So more on those microscopic and macroscopic characteristics that I mentioned. At the top there in figure A, you have Trichophyton rubrum, which is characterized by a downy white colony morphology with a deep red reverse color on agar plates. Microscopically, you can see that it produces those clavate microconidia alongside the hyphae. In spot B, you have Trichophyton mentagrophytes, which I'll go into a bit more detail about in just a sec. And then down the bottom there, you have Trichophyton tonsurans, which is characterized by its suede to powdery colony morphology with a brown complexion and a deep red reverse pigmentation. The conidia formed are of a varying size, with some cultures also producing irregularly shaped macroconidia. Brief mention to 2 species of Trichophyton that do not produce microconidia, and those being Trichophyton pseudonances and Trichophyton violaceum. So before we go into any further detail into Trichophyton indotineae, the whole reason we're here and why we have this new assay, I've just got to briefly talk about Trichophyton mentagrophytes, its background, and its relation to Trichophyton indotineae. So mentagrophytes is the second most isolated Trichophyton species that we see in humans. You can find it worldwide, and it can cause both nail, skin, and hair infections. These infections are usually acquired via animal transmission, so your pets or domesticated livestock, However, with the rise of indotineae, we are seeing an increase in human-to-human infections as well. Microscopically, you can see that Trichophyton mentagrophytes also produces those microconidia. However, they are more rounded than clavate in this species. They also produce those cigar-shaped macroconidia that are septated. And in some cultures, you produce this spiral hyphae that you can see in the bottom right. Macroscopically, they also have a powdery white complexion with a yellow to brown or deep red reverse color on the agar plates or agar slopes, depending on which your laboratory uses. Now on to Trichophyton indotineae. So for those that haven't heard of indotineae as yet, it's a highly infectious drug-resistant fungus that can cause extensive tinea infections that are difficult to treat. This difficult to treat encounter is due to the resistance to terbinafine, the first drug of choice that we have on hand for dermatophyte infections. The species has emerged from India within the last decade and has now spread globally, including to here in Australia. This spread and this new species of Trichophyton is suspected to have originated from the overuse of topical antifungal combinations that you were readily available to buy over the counter overseas. In 2020, Trichophyton indotineae was proposed to be a new species from Trichophyton mentagrophytes variant 8. After genomic sequencing of its internal transcriber spatial region of ribosomal DNA was sequenced and found to be unique, cultures of Trichophyton indotineae are almost morphologically identical to those of Trichophyton mentagrophytes, which you will see in just a minute. However, Trichophyton isolates do not usually display a positive urea culture. Typically, those Trichophyton indotineae infections respond poorly to terbinafine. A host reaction can include widespread lesions that affect multiple body sites, with these lesions being painful, itchy, and inflamed. Both immunocompromised and immunocompetent patients can be infected with the fungus, which can spread easily from person to person. Due to the severity of the symptoms that I'm about to show you, the fungus can often be misdiagnosed as some other disease. So you have that spready rash that can affect multiple body sites. It can be raised lesions that can be often bloody and inflamed. And it, again, can affect multiple body sites. So at this point of the talk, I just want to go into the characteristics of Trichophyton indotineae and its colony morphology with its microscopic and macroscopic morphology. And then I'll go into some side-by-side comparisons between it and mentagrophytes, showing why it is almost indifferential between the two species if you're looking at it in culture. So you can see that they still produce, like mentagrophytes, that powdery white colony morphology with a yellow to browny red reverse. Microscopically, you can still see those clavate microconidia, those septated macroconidia, and then some spiral hyphae can also be seen within cultures. Side by side, as you can see, Trichophyton indotineae and Trichophyton mentagrophytes are almost indistinguishable from one another. They both have that flat cottony to powdery white colony morphology with the yellow to bright brown reverse. And again, microscopically, you can see that they both produce those very similar looking microconidia, macroconidia, and spiral hyphae. The similarities, however, do not stop at the macroscopic level. If you look at the internal transcriber spatial regions of the two species, there's only a 2 base pair difference between Trichophyton indotineae and Trichophyton mentagrophytes. Similarly, there's only a 3 base pair difference between Trichophyton indotineae and Trichophyton indodigitale. So onto that resistance that you can see within the species of indotineae. There is a noticeable 2 points of resistance that can be observed. So you've got your terbinafine resistance that I've already touched upon. This is caused by point mutations in the sequentiline epoxidase gene, or SQLE. These mutations cause amino acid substitutions that alter the enzyme's active site, causing terbinafine to no longer bind to sequentiline epoxidase, rendering the first line of treatment ineffective. There's also a noticeable increase in azole resistance within the Trichophyton genus as a whole, including Trichophyton indotineae. So an overexpression of the CYP51 gene leads to an overproduction of enzymes that azoles target. This increased enzyme concentration dilutes the inhibitory effects of azoles, reducing treatment efficiency. So like your antifungals like itraconazole and voriconazole, just to give two examples. Here's a diagram depicting those two resistance mechanisms in action within a fungal cell, and how the overexpression of CYP51 and SQLE mutations can cause antifungals to be ineffective. As I mentioned earlier, it's agreed that Trichophyton indotineae has originated in India. Over the last 10 years, it's proliferated across the country and has now reached epidemic level there. Around 78% of all dermatophyte infections that are encountered within India are Trichophyton indotineae. Over the last 10 years as well, it has spread throughout Asia and has made its way to Europe, the Americas, and here in Australia. There's also a noticeable increase in the level of the species in Germany and the United Kingdom, as you can see on this heat map, with the darker shade of blue compared to other Western countries like America and Australia. So after all that information on indotineae, to summarize, why do we need a new assay with a gene target for the species? So as you've seen, indotineae is morphologically very similar to Trichophyton mentagrophytes. Currently, assays on the market cannot differentiate between indotineae and mentagrophytes. There's a public health concern around the species due to its terbinafine resistance and the spread that's happened rapidly over the last decade. Currently, the gold standard for identifying a Trichophyton indotineae infection is DNA sequencing. However, this can be expensive and have a longer turnaround time than a commercial PCR assay. Currently, there's an unknown scope or current levels of Trichophyton indotineae in the community. And there is a lack of education within the medical community to the rise of indotineae and its spread. Now, there's a lot of information on this table. I just want to highlight a couple of things on the different methods of indotineae infection. So with your culturing, the cultures can take up to 1 to 3 weeks. However, due to its similarity with Trichophyton mentagrophytes, we can't use culture as a foolproof way to identify a Trichophyton indotineae infection. Leaving us with genome sequencing as the gold standard, like I said. However, this requires high caliber bioinformatics processing and reference databases that are quite expensive, which is impractical for public and private pathology laboratories. Moving on to the second half of this talk, I just want to give a brief overview of how the R-Biopharm PCR assays work, and then onto the work we've done validating the new dermatophytes A assay that contains the target for indotineae. You currently can't find this target on the dermatophytes and other fungi 12-well assay that is on the market. This will include how we perform the validation and the findings that we found. So R-Biopharm assays use a multiplex tandem PCR, or MT-PCR, to allow for the detection of multiple gene targets in one sample. This allows for the lowest quantity of nucleic acids to be amplified, even while mixed in with other higher concentrations of other genetic material. You can use as little as 10 microliters a sample, leaving you with more to use on other tests. And the technology uses nested primers, which are used to ensure only the target nucleic acids are amplified for detection in the assays. Each target is amplified independently, which eliminates competition between each target. So MT-PCR is a two-step process, which I'll refer to as step 1 and step 2. So in step 1, a set of outer primers, both a forward and a reverse, are used to amplify a broad range of target DNA. This short 15-cycle multiplex preamplification reaction uses the primers that are homologous to all targets in the panel. In step 2, a portion of the amplified DNA from the first round is used as a template for a second round PCR reaction. This second reaction uses a different pair of inner primers that anneal to a site located within the DNA fragment amplified in the first round. The PCR reaction is performed in the MT analyzer, where the DNA amplification is measured by the increased fluorescence of Ava green dye, which gets incorporated into the DNA being formed in the specific amplification reaction. This nested approach increases specificity because the second inner primer will only bind and amplify the target region if it is correctly amplified by the outer primers in the first round. The increased total cycle number also enhances the sensitivity, making it possible to even detect small amounts of DNA. So like Misha said, this is the current dermatophytes and other fungi 12 assay that's on the market. It has all your Trichophyton species gene targets, including rubrum and mentagrophytes complex, which includes Trichophyton indotineae, microsporum species and microsporum canis, epidermophyte flocosum, and Nonazia gypsia. So the new dermatophytes A12 assay still contains all those Trichophyton gene targets with the addition of Trichophyton indotineae. Some of those gene targets for the candidas have been removed, with only albicans remaining, and the Aspergillus species gene target has also been removed. So with that look at what gene targets are currently present on the dermatophytes A12 assay, what was the aim of the validation that we did on behalf of R-Biopharm? So the main aim was to validate the new dermatophytes A12 assay for detection and correct identification of Trichophyton indotineae from Trichophyton mentagrophytes. To do this, we had collected 508 samples, with 472 of those being clinical in nature, either nail, skin, or hair, with 36 direct fungal cultures also used within the validation. 30 of those direct cultures were confirmed Trichophyton indotineae isolates provided by the National Mycology Reference Centre over in Adelaide. So to do this, we first had to prepare a lysis buffer preparation, which in this case was the prep tissue digestion kit buffer. The buffer comes in two parts, buffer A and buffer B, which are just combined into one lysis solution. That solution was pipeted out into PCR tubes with sample added to them. The samples were then heated at 70 degrees for 30 minutes and centrifuged down. 200 microliters of this centrifuge eluate was then headed into a 96-well extraction cartridge and run on our Roche MagnaPure, which is our extractor of choice here at St. Vincent's. The extracted elution then underwent a step 1 and step 2 PCR reaction on R-Biopharm's automated amplification machines, using both the Derm A 12-well panel and the dermatophytes and other fungi 12-well assays in parallel. Results were then generated by the analyzers. So of the 508 samples tested, roughly 43% were toenail clippings, with a further 25% being nail clippings of unknown origin, or where the site wasn't stated, I should say. The remainder 32% were miscellaneous assortment of skin scrapings and hair, with some of those skin scrapings being mostly from legs, foot, groins, and some scrapings. And then 7% was also those direct cultures that I mentioned that we had on hand. So what did we find when we ran these two assays in parallel? First, on to the dermatophytes A12 assay results. So of the 508 samples that we ran in parallel, roughly 240 samples were positive for Trichophyton rubrum, which isn't really anything outrageous, given that it's the highest turnaround of the species that we see within our laboratory. Around 60% were positive for Trichophyton mentagrophyte, or 60 were positive for Trichophyton mentagrophyte, I should say, sorry. Just under 35 were positive for Trichophyton indotineae, and then we had roughly 150 negatives. And there was a small amount of epidermophytum flocosum, Nunnersea aegyptia, and Microsporum canis that were also picked up on the assay. When we compare these results to those on the dermatophyte and other fungi assay run in parallel, you can see that the results between the two assays almost matched up perfectly. There was a larger than usual gap between the positive Trichophyton mentagrophytes in between the two assays, however. This isn't due to any discordant result, but due to some of those samples that were called Trichophyton mentagrophytes on the derms and other fungi assay actually being called Trichophyton indotineae on the new dermatophytes A 12-well assay. I'll go more into that in just a sec. All 30 samples that were known Trichophyton indotineae from the National Mycology Reference Center were also correctly called Trichophyton indotineae on the assay, which is a big thumbs up from me. Overall, 97% of the samples were concordant when we ran them in parallel. There were only 12 samples out of the 508 that were tested for gene targets that were discordant. These discrepancies may have been due to some sampling issues, with retesting at the time unable to be performed due to insufficient sample being left. Some of the discrepant results may also be due to some DNA degradation after retrieval from long-term storage. We were collecting these samples for well over a year before the validation happened, so it's understandable that some degradation may have occurred. So now I'm just going to touch on those samples that were called Trichophyton indotineae on the new dermatophytes A 12-well assay, but were called Trichophyton mentag on the derms and other fungi assay. Currently, within our laboratory, we are relying on doctor requests and clinical notes to test and rule out Trichophyton indotineae infections. So whenever we have a request slip that comes into the laboratory with a query Trichophyton indotineae or a resistant fungal infection, we set up a culture, which is not something we do routinely anymore since we moved to the dermatophytes PCR assay to identify all our dermatophyte infections. During this validation, we were lucky enough to have quite a number of patient samples come within the through the laboratory with slips like this. So running these samples on our existing dermatophytes and other fungi assay, we had a result of the samples being positive for Trichophyton mentag with positive Blancophore as well. Cultures that were set up on these samples also grew what looked to be a Trichophyton mentagrophytes complex. However, when we ran these samples on the dermatophytes A assay, the results tell a different story. So as you can see, the assay has called these species to be a Trichophyton indotineae and not a Trichophyton mentagrophytes. Now on the screen you can see that the gene targets for both Trichophyton species, Trichophyton mentagrophytes, and indotineae have all gone positive. So I spoke earlier on how genetically closely related mentagrophytes and indotineae are. The assay is going to, at least when we were validating it, pick up any species with indotineae also having mentagrophytes. There is, however, on the panel a reference gene that in the background in the programming will call a species either Trichophyton indotineae or Trichophyton mentagrophytes. And in our case, it called them Trichophyton indotineae. With this result, however, we we cannot obviously report this out to the patient, given that this was currently an unapproved assay via the TGA. So in the meantime, the cultures that we had grown that looked like a Trichophyton mentagrophytes, we sent off to the National Mycology Reference Center for genomic sequencing. And a few weeks later, we got those results back, identifying that those samples were correctly a Trichophyton indotineae. So seeing those the indotineaes match up on the new dermatophytes A assay and through genomic sequencing was a big win and shows that the assay can successfully differentiate indotineae from Trichophyton mentagrophytes. So we know the assay works. We successfully completed the study's aim for determining that the dermatophytes A assay could detect and correctly identify Trichophyton indotineae from Trichophyton mentagrophytes. What's it going to bring to the table in regards to patient care and laboratory implementation? So culture and microscopy have always been our gold standard for identifying dermatophyte infections. Though these can take a few weeks to identify a species, when you look at their whole morphological profile, you could generally get a reliable identification using this method. With the onset of antifungal resistance in some dermatophytes and their changes at a species level only really showing up at a genomic level, change is needed to our testing practices. As we have seen, indotineae is morphologically and genetically similar to Trichophyton mentagrophytes. Its infections can cause a much more severe reaction in patients, so a rapid identification is needed to enact correct treatment outcomes and minimize the spread of Trichophyton indotineae. With this new dermatophytes A assay, these outcomes can be achieved. It'll give you a rapid result within 4 to 6 hours, depending on your laboratory's workflow, and it will differentiate indotineae from mentag. It will stop the need for genetic sequencing and the impact of this increased workload has on our friends at the reference laboratories, while also giving us a more accurate level of Trichophyton indotineae currently found within the population. So like I said, the assay will give you a rapid result within 4 to 6 hours. This result will allow the rapid identification needed for clinical staff to give feedback of the identification to requesting doctors and consult on treatment options and patient care. Patients having this result so rapidly will allow them to minimize the spread with close family and friend contacts. In your laboratory, using this assay will minimize the number of culture media needed to make a presumptive identification of a possible Trichophyton indotineae. It'll minimize the time spent working on the specimen, allowing for staff to be redeployed to other tasks that need to be done within the lab. And it'll minimize the number of isolates being sent to reference laboratories for genetic sequencing. So, in conclusion, the R-Biopharm dermatophyte assay is a reliable method for diagnosing Trichophyton or indotineae from Trichophyton mentagrophytes, as well as all your other dermatophyte infections on skin and nail samples. It enables a fast diagnosis without the need for genomic sequencing or culture results. It enables faster treatment, improving patient care and reducing laboratory burden. And what I think will hopefully be the big takeaway from using this assay, we'll get a more substantial population level monitoring of Trichophyton indotineae that is currently being overlooked, missed, or misdiagnosed as Trichophyton mentagrophytes. Thanks, guys.
Thanks, Josh. That was great. Thank you so much for your presentation. I'm just going to open up the floor now for questions. So you can ask questions via the Q&A button at the very top of your Teams chat. I'm just going to go ahead and go straight into it. So the first question is, what robots can you run the assay on? This is currently compatible with both our low to medium throughput device, the Rita Explorer, and then also our high throughput device, the Rita Jump. So you can run it on either assay. Josh, there's an interesting one here. Do you have a breakdown of the sites from where Trichophyton indotineae was detected? So do you have like a kind of distribution between the skin, hair, and nail specimens that indotineae was seen in?
The ones that we had in the laboratory were all skin, I believe. We didn't have any toenail ones at the time. Not to say that it can't infect toenails, but it seems like skin scrapings are the go-to at the minute for an infection.
Yeah, I guess that's just also with the nature of transmission, hey, that you're probably more likely to see it within skin specimens than you are. Is that true, or?
Yeah, I think it's just generally how people are close. They want to be close to each other. We spend time with our friends, we spend time with our family. And unfortunately, sometimes you just happen to rub up against someone, and that's how easy an indotineae infection can occur. So, yeah, seeing it in a skin sample is really not that strange, I guess.
Yeah, okay. Hopefully that answered that question. There's an additional one here. Did the assay detect any additional targets in your verification? If so, what percentage of samples in your cohort did this occur? I don't think that we have had any additional targets that were detected or off-target detections as part of this assay. I think it was quite specific, but Josh, if you also want to comment on that.
Is that in regards to some of the targets that you find on the old assay as well as the new assay? Because in our current dermatophytes and other fungi 12 assay, we do have the targets for all the candidas and aspergillus and scopulariopsis turned off. So we didn't have those, obviously the candida and scopulariopsis turned on in the when we were validating the dermatophytes A assay. If that answers that question. Unless, Darren, you want to say something?
No, well, I'm just trying to get my head around the question. Sorry. Look, I don't think we actually found anything when we were testing the targets in parallel. We really didn't find anything extra that we weren't expecting. I mean, there were some of the discordance where we did find, a ruben and in one and not in the other, but that's again something we found very early on when we actually started. We started doing this testing about 6 years ago, and the samples themselves can be very prone to sampling. When particularly if you're dealing with nails, you're clipping off a bit of nail. It's very hard to get something that's uniformly distributed. But no, I don't think we actually found anything that we weren't really expecting.
Yeah, I mean, I think Josh also, you mentioned that you did also touch on that there was a couple of samples that were positive previously for mentagrophytes that were then positive for indotineae.
Yep. Yeah, so those ones they still matched up. So in the derms and other fungi assay, they were called Trichophyton mentagrophytes, but then when we tested them on the derms A, they were indotineae. So even though one called it indotineae, one called it mentagrophytes, we're still going with it actually being an indotineae and not like a discordant result, because that's what the assay is here to do. It's meant to tell us if it's a mentag or an indotineae.
Yep, I have a more technical question here, also in relation to Trichophyton indotineae. Since there's only a difference of 2 to 3 base pairs between the indotineae and the mentagrophytes, how reliable is the assay to differentiate the species? Are there any possibilities for misdetection? Certainly from our development perspective, we did put a lot of time in making sure that the primers that we have in-house were definitely specific for indotineae, but also what did you observe, Josh? Do you think that there was anything unexpected in terms of any as there was 100% correlation between indotineae, but any other comments that you wanted to make on this specific question?
No, like I said, we had those indotineae isolates from the National Mycology Reference Center, and given that the assay was able to successfully identify indotineae in all 30 of those samples, I don't think there's a high chance that you're going to see any misdiagnosis. But that's what we found anyway. I can't say what the future holds for other people, or if something goes wrong, or what may happen.
Or if there's any sort of genetic changes and things over time. But I mean, given what we've seen, we had some highly characterized samples, plus those mentags that we picked up as mentags that then turned out to be indotineae. We didn't see any evidence of that.
Thank you both. I think that was definitely answered that question. There's an additional one here. With the dermatophytes A panel released, what is the benefit in still using the old panel? Or if you guys have any additional comments, but at least from the R-Biopharm perspective, with the dermatophytes and other fungi, we do note that it's marked as IVD in the A and Z UK and EU regions. So there's still that benefit. But I think this definitely has a lot more clinical utility. It can definitely help more in targeted antifungal treatment. What are your guys' thoughts on this specific question?
As soon as we can, we're switching over, and we will no longer be using the old panel. So yeah, so we're just running down our stocks of the other one and just start. So we'll go live in the next few weeks.
Yeah. And I'll be doing a nice little happy dance when that happens.
Yes, Josh is very keen.
I can understand that.
Mm-hmm.
Just a couple, another one here. Have there been any changes made to any of the other targets that were on the previous panel and still on this panel? The only change that we made was in the Microsporum canis, just tweaked it a little bit so that it performed a little better, not with any adverse effects. I think we still picked up 100% of the canis. But other than that, there were no other changes made to any of the other targets. So it is still the same primer sequences for most of the other for all the other targets that are shared, like the epidermophyte flocosum or the Nenetzia gypsia. There's another question here about is this panel compatible with FFPE sample types? Josh, I don't think we ran any FFPE sample types. I don't know how common it is for us to be collecting that sample type in relation to dermatophytosis or onychomycosis.
I don't think it would be used. We definitely didn't use any within the validation. I think it's for your skin. Yeah, it's for your nail, your skin, and your hair.
Yeah, I imagine that FFPE is perhaps not necessarily required when you're collecting something so fresh as well from a patient and then putting it directly into the extraction reagent.
Yeah, so like those samples, when we get them here in the laboratory, they're only sitting, they come into our specimen reception, they're within the laboratory within half an hour, and then we do a big dump at the end of the day in processing. So they're only sitting around for an hour or two at most before they're processed and ready to go on the assay. So yeah.
Yeah, there's a question here. For the samples that you tested, Josh, did you also perform direct microscopy? And if so, how did those results correlate with culture and PCR?
Could you repeat that, Misha? You broke up a bit there.
Oh, sorry. For the samples that you tested, did you also perform direct microscopy? And if so, how well did those results correlate with culture and PCR?
I suppose the one thing is oh, sorry, Josh.
No, you go, it's fine.
I was going to say, the one thing to actually say was our standard protocol is to do direct microscopy on everything. So we do blanket stains on all of our dermatophyte samples. So we've moved from doing a blanket stain and culture to doing the blanket. So we do microscopy on every specimen that's actually received, plus the PCR. And if we see something if the PCR is negative for the targets we test, but there is something that we see by microscopy, we then reflex on do culture. So there is some culture that we still do. Now, most of the time, that's probably around about 5% of our samples go on for culture. And it's pretty much most of the time, it's really mostly things like aspergillus or some other things that we sort of candidates that we pick up that we're not actually testing for by the PCR. Interestingly so, but on the whole, microscopy and the PCR does marry up very well. Occasionally you will see things by PCR that you don't necessarily see by microscopy. And interesting, it tends to be when that happens, it's the mentag groups. For some reason, the mentag doesn't necessarily, we seem to pick it up better by the PCR than we do sometimes always by microscopy. But on the whole, microscopy and PCR are pretty concordant.
I mean, sometimes you're always going to get a nice big juicy tub of toenail clippings where the patient's been collecting them for a few weeks. And it's not a nice image to picture, but sometimes when you get all those toenails or with all that skin scraping, some sampling issues can occur. Like you can get a bit on your blunkful that's positive, and then a bit that's being used for the PCR assay isn't actually infected, and vice versa. But that's a very low margin.
That's interesting. Didn't realize people were collecting infectious toenails or nails.
Oh, you have no idea.
I'm just glad that Josh is not actually showing photos.
Looks like we've got a couple more questions if you'd like to finish them, Misha, and then we'll do a closing.
Yeah. So just really quickly, what other extraction reagents can the tissue digestion buffer be used with? So currently it's validated for the R-Biopharm portfolio. So that's the Rita Extract 24 pathogen kit, and then also the Rita Extract 96 kit. And an additional question here, how many samples can be run simultaneously? On the Rita Explore, that's 24 samples, and then on the Rita Extract 96, that's 96 samples simultaneously. Being mindful that because of the pre-digestion step, there is a limitation there with a little bit of manual handling, but once it's put onto the robot, it's 24 samples on the Explore or 96 on the Jump. And for any pricing queries, please refer to your account manager. They are best poised to help you, with any questions that you might have about cost or otherwise.
Oh, Libya, you're on mute.
Thank you. Darren, would you just like to do some key takeaways for everyone who's still online?
Yeah, look, we moved to molecular detection of dermatophytes probably around 6 or 7 years ago now. It's made a dramatic difference to us, both, I suppose, as the clinical service we're providing. So, you know, we shaved turnaround time for dermatophyte reporting was usually on average 3 to 4 weeks. Now it's usually 3 to 4 days. So with that encompassing both the Blanket stain and the PCR result. But also operationally, it's made a massive difference to the laboratory. Our mycology lab has really gone from a full day down to a half day. So now we're actually utilizing that EFT for other areas, because we've been able to really make a lot of time savings rather than people having just sitting there looking at dermatophyte cultures day in, day out. So we've effectively saved the laboratory probably 3 to 4 hours a day by actually moving to dermatophytes. So again, it's not just, I think, to the benefits of the clinicians. There's definitely benefits to the laboratory and actually taking this up.
Thank you so much, Darren. That was really great. A really good summary. I'd really like to take the time to thank everyone for joining us on today's webinar. We will follow up with a post-webinar email with some of the highlights, but also that recording if you'd like to watch it back or share it with your colleagues who weren't able to make it. I'd like to thank Joshua for your research and your presentation, and Darren, to you and your entire team at St. Vincent's, a big thank you for your support and being online with us today as well. And Misha, also thank you for joining. Misha is a great point of contact if you would like to follow up with anything technical. Otherwise, please don't hesitate to reach out to us via any means of communication, and we'll make sure that we get back to you. Thanks again for joining, and have a great rest of your Tuesday.